Let's do a little review real quick. My carbohydrates. What elements do I have?
H. Cho.
I had CHO.
Yeah.
Lipid. What do I have? What do you guys see?
John.
I never saw any nitrogens in it.
Oh.
Look at it again. Do you see any nitrogens?
No.
No, it's CHO. Now, how would I be able to tell the difference between carbohydrates and lipids? Because I said there's something very specific about one of them.
Carbohydrates have a ratio of 1 to 2 to 1.
We have a 1 to 1 ratio. Now, with my lipids, this is many. And this one is few. That's about as simple terms as you're going to get. There's buttloads of these CH groups, and there's just a few hydrogen or a few oxygens hanging out. So now we're going to talk about proteins. Okay? Let's talk about our proteins. Carbohydrates, quick sugar. Liquids, they do a few things. Proteins are insane. They also have 6 things, but there's so many of them out there. First off, before you write anything, let's get the important stuff. Proteins are also known as polypeptides. Proteins are also known as polypeptides. I use those 2 words interchangeably. Sometimes your worksheet would say a polypeptide. Sometimes it will say a protein. Sometimes it even says an amino acid chain, because the amino acids are your monomer. So we take amino acids and we build them together to make the polymer, which is your protein. How many amino acids does it say there are? 20. Yes, ma'am. What was your question?
So it's a building block, really?
This is the building block, and it builds proteins. Now, I told you earlier, I don't care about any of those silly little bond names or anything like that. I only care about the hydrogen bond. I lied. The only other bond I care about is the peptide bond. I'm not even going to teach you what it means to be a peptide bond, but I'm going to teach you the reason why I care about it. All your little amino acids are held together by a peptide bond. And that's why proteins are known as polypeptides. It stands for many peptide bonds. That's why I like science. It's simple like that. We use big words to mean simple things. Now it says we have 20 different kinds of AA. That's not Alcoholics Anonymous. This is a different AA. The little a means amino acid. And I don't usually write amino acids anymore. I just write little a, which means now you have that opportunity just to write little a to mean amino acid, as long as you understand it. It says there are 20 different kinds. Think of them like the alphabet. If we rearrange the order of them, add extra, take away some, we can have lots of different proteins. Anywhere between 1,000 to 10,000 proteins. And it's just different combinations of amino acids. There are proteins that do everything. There's a protein to give you freckles. There's a protein to give you blue eyes or brown eyes. There's a protein to give you red hair, blonde hair, curly hair, straight hair, no hair. Right? There's proteins for everything. This is their basic jobs. There are proteins that will store stuff. We can even store a little bit of energy in them. All right? We have proteins that can transport items throughout your body. The example they give is hemoglobin. Have you heard of it before?
Yes.
Where?
I feel like it's in your blood.
It's in your blood. The hemoglobin actually transports oxygen and carbon dioxide. So if you have more hemoglobin, you can have more oxygen, which actually can make you a better athlete.
I wish I want more hemoglobin. All the time I just feel lightheaded, even now.
Less hemoglobin.
Yeah, so my running is
Or less exchange of it. You may have lots of it, but it may not work properly. There's regulatory, which means they can basically send messages, like we said with the lipid hormones. Well, we have protein hormones. Like when scared y'all earlier. You started having a little bit of adrenaline running through your system. It helps with reflexes. That adrenaline will help. There's other hormones that are sent by different parts of your brain throughout your whole body to help say, hey, guess what? You're a little hot. You should sweat now. You're a little cold. Commence shivering. Alright? There's lots of little weird hormones running through you. Not like the hormones yesterday when we said, oh yeah, look at that hot one going down the hall. That's a different hormone, alright? We have structural proteins that help build stuff, like muscles for movement. We have membranes. We've got hair, nails. Your skin is a huge amount of proteins.
Yeah.
Alright? Proteins are all over the place. And then the one we will specifically look at tomorrow and definitely on Friday are enzymes. Because they help control how fast or how slow chemical reactions happen in your body. Like Friday. We're going to use a very specific chemical that you guys have in your house. It's in a small brown bottle.
Peroxide.
Hydrogen peroxide.
Yeah, it's like that.
And when you get cut and your mom dabs a little bit of hydrogen peroxide onto a cotton ball and puts it on. Oh, she pours it.
Yeah,
Oh, you got the nice mom.
My mom pours
That's
it.
what I do.
Okay, so what happens?
It bubbles.
Okay, so we got bubbles.
It hurts.
And it burns because, you know, we got that pain.
Real bad.
You guys learned back in junior high, anytime you have the formation of bubbles, or the formation of gas, or there's heat, it was a chemical reaction. Well, guess what? The hydrogen peroxide is not the enzyme. There's an enzyme in you that made this happen. So we're going to do it with liver. And we're going to test enzyme action with it to see what happens, okay? I love liver, it's good stuff. Yes, ma'am.
Can we bring Vicks and put like?
It won't smell bad. Actually, you have to smell part of it. I'm not sure why y'all are all worried. I'm in here for like 4 periods.
We did it in Reed in like 5th grade.
We
Oh, yeah.
mummified chicken livers.
Yeah. it
No,
was
we're not
so
mummifying
bad. They were
anything.
just
We're not
so
mummifying.
bad.
Actually, the
I
amount
still
of liver
have
you
mine.
get,
Why?
the amount of liver you get is but the size of your thumbnail. That's all you're getting. That's it.
Oh, because ours were like the size of
We
a
did.
dog.
Yeah, but we're not mummifying it.
Yeah.
We're destroying it. Possibly. We don't know. All right. Next. I said next. Hello, cursey thingy. Thank you. Guess what? I don't want you to write that down. I know, right? You're very happy. Let me explain. So, carbohydrates, what do they look like? Ring. Okay. Lipids, what do they look like? That's right, gang symbols. Okay, so tails, right? So, peace. He now's the peace symbol. Okay, anyway. Proteins are slightly different. They actually have to go through 4 different structures to be a protein. There
So,
are
like,
4
they
different phases.
metabolize.
It does, actually. Now, this part you're going to write down. And you've already written part of it. It's made of amino acids held by peptide bonds. Now you just add that they're in a straight line. They're in a long, straight chain. And it does say amino acid 1, amino acid 2, 3, and all that. It could be, you know, amino acid 1, amino acid 1, amino acid 1, amino acid 4, amino acid 18. Again, there's 20 of them. Okay? But first off, they're in a long, straight chain.
That's what it looks like. Okay.
So each one of these is just the, it's not the carbohydrates. This is just amino acids. The amino acids are weird looking to begin with. And with them being 20 of them, we're not asking you to recognize each individual amino acid. That's not going to happen. But we have a long chain of amino acids. So now, look what happens on the second structure. What do you see happening? This is still a chain.
They corkscrew.
Okay, so they twist, or
They
zigzag. They chevron.
they'll fold, right? This class today is all about like hand movements. Okay? So either they're going to twist, or they start to fold. So this was that long chain. It twisted or folded. And then here's the third structure.
What is that?
First period said, Mr. Nielsen, did you let like one of the kindergartners come up here and scribble on your board?
I want to ask you to draw that.
I did not draw this. This is done by Steve Messingale, who we borrowed the PowerPoint from. But I still see a coil. Okay, so it twisted. I still see these folds. And then
Scribbles.
we have the scribbles. All right? But that's just the third structure. How many structures do we have?
Can we draw those though?
I'm going to show you how to draw it.
Oh.
Here's the fourth structure.
So they split the fourth and
No, there's 4 of them together.
Oh.
There can be 18 of them together. There can be 2 of them together. There can be just one. But they have the ability to build together. This right here is an entire protein. What was the monomer? One what? Amino acid.
Yeah.
And then we put a whole bunch of them together. And then we put those guys together to make proteins. Okay, so here is the polypeptide. Here is our polymer. Now, really quickly, we said ring shape for carbs, long tails for lipids. What is the highly scientific term for the shape of proteins? It's on here.
Spaghetti.
It's on here.
Subunits.
Globular.
Okay.
Globular. The scientific term for the shape of proteins is globular.
So glob, because it's like
Very
a glob.
science.
It looks like, you know, a piece of bubble gum or something that somebody took out of their nose.
Okay.
All right?
Uh-huh.
It's globular. It's not ring, it's not tails, it's globular. So, let me explain what you would write down. Remember you had that long chain?
Yes.
Okay, so this is how you form a protein. That long chain will twist and fold. There you go. Some people figured it out. It will twist and fold. It will twist and fold.
Wait, what?
Oh, believe me, there's more dancing with hands later. You guys get to follow along with it.
It took me a minute to realize what you were saying. Yeah.
It will twist and fold into a complex. Because believe me, that's complex, is it not? It's not like simple ring shape or the gang symbol or anything like that. It will twist and fold into a complex but specific structure. It will twist and fold into a complex but specific structure. And then sometimes you can put those structures together to make another protein, okay? And it is known as being globular in shape.
Complex but specific.
Into a complex but specific shape. Every protein is very specific in its design. Okay? Every protein has a very different shape from every other protein. Now, really quickly,
They are polar. They form in water.
what elements would we find in this?
Show.
Cho. Gotta have cho.
Gotta have.
Are you going to have Chan?
We are gonna have chon. Puh. Alright, so carbon, hydrogen,
Oxygen, nitrogen, something.
phosphorus,
And
and sulfur. Alright? Doesn't always have those, but for the most part, those are the elements you find in your proteins. And now it sounds like you having a stroke when you're trying to say it. It's chon.
Chom.
Because you want to say chomp, but it's and then chon.
Wait, are you going to give us like the ratio or like?
There are no ratios.
Because they're all evenly distributed?
No, they're all totally different every single time. Every single one of them is slightly different.
So
So it's like a snowflake. So you
Okay.
just
Kind of like that. Yeah, I like that analogy.
I think it's not every time I do it.
Like a
Wait, what'd you
what?
say?
Like a snowflake.
Snowflakes.
Okay, last stuff. The nucleic acids, which you've already discussed. What are the 2 examples of nucleic acids? deoxyribonucleic acid and ribonucleic acid, which you guys also known as DNA, RNA. Hey, guess what? If you already knew that, why are you writing it down again? Right?
Good question.
Nucleic acid, RNA, DNA. RNA's got that double helix, so it's that twisted ladder. RNA is just a single strand, so it looks like a cone, a very straight cone. The new stuff you have to add, though, these are their polymers. How do you build them? You use
Neat. I don't even know how to say that.
the monomer.
Wait,
Nucleotide. Nucleotide.
wait.
Nucleotide.
So if the RNA is a kind of what is the DNA? Of what shape?
The DNA has two sides. RNA only has one side. There's a few other differences, but we'll go more in depth with that later. And again, just like everything else, if we want to put these nucleotides together, just like if we want to put any other monomers together, we use dehydration synthesis, which means what's going to happen?
Like lose the water.
They'll lose water. We remove the water to allow them to join together. Simplistic. It truly is. We get to figure out why I have this drawn in my hand now.
Yeah, I've been wondering.
I know you guys have all been wondering, why does Nielsen look like an idiot? Very specific reasons why I look like an idiot. See, look at that.
Big difference.
No, not that part, just part. All right. I had to show the viewers at home. All
I
right.
wish we were on like a real TV show.
We are
Yes.
What's it called?
live with Nielsen. All right. It's like Regis and Kathleen, but it's not. So, really quickly, this is what your nucleotide is made up of.
We need
These three items. So, phosphate. Yes, we're going to write them. What element is going to be found in phosphate? Phosphorus. So we're going to find something with a P. Actually, there's also oxygens in there too, so that's right. Pentose, what is this? It's a sugar. We know it's a sugar because duh, they said it's a sugar. But what's the other reason why we know it's a sugar?
It ends in O's.
It ends in O's. So what's the pent?
5.
5. So how many sides to my ring would I have? I'm going to look for something that looks like a pentagon. A really ugly pentagon. There we go.
Aw, it looks like
I just spelled sugar as H-U-G.
I spelled it S-U-P-A-R.
I don't know why I did that.
I really get my letters confused a lot.
I'm noticing this. And then last but not least are your nitrogenous bases. So there will be nitrogens in them.
Good job.
Adenine, thymine, uracil, cytosine, and guanine. You guys remember talking about A's, T's, G's, and C's?
Yes.
You don't have to remember everything about it, because I'll help you with that. But you do remember talking about A's and T's go together, G's and C's go together. And you might have talked a little bit about RNA, in which case then we don't have a T, we have a U. And again, if you don't, that's okay. I get to learn, I get to learn you. I get to learn you.
Will notes ever get easier?
D9.
Now, but what we're going to do
Don't skip this.
is next week, we're not really taking notes. We're going to put this all into practice.
Yay.
So the notes are helpful just to get the just to get the basic knowledge. We're going to do different activities, different worksheets to help you understand them better.
Don't skip it.
If I was to do the activity first, you'd have no clue as to why. All right? Remember when we did Baby Dice Island? You had no clue why we were rolling dice and stuff like that? But afterwards, we started talking about the J curves and the S curves.
Yeah.
Well, this week's lab, we're going to reverse it. I'm going to tell you about enzymes, and then you're going to see them happen on Friday. Next week, we're going to take these notes about, biomolecules, and we're going to see if we can't make it a little bit more understandable for you. Cool? So, you guys ready to draw what this bad boy looks like?
I have no.
It looks like this.
It actually looks like this.
I wish you got it on camera.
It looks like this. There we go. All right.
Wait.
Can't wait too much longer. We're gonna have lunch soon.
Okay, have no room, I really do.
Okay, turn the page.
No, I mean it.
Then don't turn the page. Draw this exactly as you see it.
Exactly.
Because after this, you won't have to draw it exactly like this anymore.
Yay.
But you have to understand it first. I was always told by my math teacher that there's always a shortcut to all these little math problems and all that. But you won't understand the shortcut unless I teach you the long way first. When you guys start driving, your parents are going to show you how to get to different places. And then you're going to find shortcuts to get there faster. Okay?
Oh, a circle. I know how to draw that.
So, oh yeah, it is pretty good. So here's my phosphate group. How do you notice the phosphate group?
Because it has a P.
We have that P in there, right? And then we have some O's.
And that is the.
What you don't see is there's on the end of this O's an oxygen, on the end of this O's an oxygen. That's it. Then, what do we have right here? It's a pentagon.
It's pentose.
It's pentose, okay? This is my sugar right here. Here's my sugar ring. Now, this also looks like a ring. Well, why is it not a carbohydrate? Why is it not a sugar?
It has nitrogen.
Exactly. It has a nitrogen right here. This is what we call our nitrogenous base. A's, T's, G's, C's, and U.
And me.
And you. That was pretty good. Okay. Now, you'll notice that on these carbons here, they number them. We don't have to understand the numbers until we discuss DNA in depth. That's a couple units from now. But they will help you figure out the next slide. By the way, this is the last slide you have to draw anything on.
Yes.
The next one is all hand movements.
Yay! Is that too supposed to be a subscript?
It could be a subscript. Actually, it's supposed to be a superscript. These are superscripts. That too should be a subscript, yes. So it should say CH little t.
What about that 5?
The red numbers are actually a superscript. It just these are the numbers. So this is actually carbon number 1, carbon
Like
number
an
2,
exponent.
carbon number 3, carbon 4, and carbon number 5. Okay?
Wait, did you say CHU was a subscript?
That 2 right there should be a subscript, yeah.
Okay.
I know, there's ugly glare all over this thing, isn't there?
Kind of.
There's nothing really much I can do about it,
No, now I'm
unfortunately.
sitting over here. It's better. Over there.
If you're straight in line with the projector, glare right there in the middle.
That's what happened to me. I hate it.
There's I don't have one glare there on the edge. Yeah. We're bright like the sun.
I don't have a single glare. Oh,
Because you're at a different angle from it, which is great. These people over here don't really have much of a glare. If you're closer to the middle.
It's the same as the sun. See the sun, the equator gets the most of it, and then the whole
Yeah, exactly.
Okay, so. Do we need to write the sugar for food? Just in case.
Just so that you know that's the sugar.
Wait, why?
Okay,
Oh my god.
ready for hand movements?
No.
Yay!
I
Does
love
that
this
mean
part.
we're done with notes?
Kind of, sort of, almost. Watch this real quick. Ready? Put your hand out like this.
I like this.
Now, you notice that I've drawn on my hand because I'm a special like that.
Yeah.
What part is this?
The
pentagon.
That's the pentagon, which is our sugar. What's on my thumb?
The circle.
Is my phosphate group. So I have my phosphorus up here. And then my fingers, nitrogenous base. Now, you can do the same thing over here on your other hand, right? So here is my sugar, phosphate, nitrogen base. Okay? So I've got 2 nucleotides.
4 letters.
Each one of these is just a monomer. I'm going to try to put them together to make a polymer. Just like that.
Oh, we don't know, dude.
No.
No, it's not that bad. Watch. Look at this.
That's a lot to draw.
I told you you don't have to draw it.
Oh.
But now that you asked, now you have to.
What?
Oh, sorry. What is this guy that I just circled? It's on my hand, right? So this would be my nucleotide, exactly. I have another nucleotide right down here. I have a nucleotide over here. I have a nucleotide over here. What do you notice about the left-hand side versus the right-hand side?
There's different colors.
We do have different colors. So the G matches up with the C.
And they go like this.
Oh, they have those.
Oh, they're beautiful. That one has the like bent leg, and that one has a straight.
Oh, wait a
Keep going.
Amazing, Anna.
Keep going.
That one is saturated.
That one's up.
No. This is not
These
the.
are
This is not your saturated and unsaturated fat.
It's upside down.
Oh, one's upside down, exactly. So try it like this. Here's my two nucleotides, right? Here's my two
You
nucleotides.
got this.
Try to put them together. You can't hold at all. You look like you're slapping yourself. But if you flip it, they're holding hands, right? So as you can see right here, my phosphates are pointing up. And then over here, this one's going to hurt. Hold on. The phosphate is pointing down. Okay? That's the only way they fit, is if they are upside down from each other.
Why would you carry a shame?
So this side will always have the phosphates pointing up. This side over here, in order to actually hold on with these lovely little hydrogen bonds, which, can you pull your hands apart?
No. Well, yes.
Yeah.
You can. If you put a little force into it, you can pull them apart, which is good because we said the hydrogen bonds were weak. They're strong enough to hold our DNA together, but we can split them apart when we have to make a copy of them. It all starts to go together. Okay? Now, we will talk about this more later. This becomes very important when we discuss DNA. Wait, there's one last thing. Always is.
Have a good weekend. No school first.
We have to figure out what elements are there. We had Cho, 1, 2, 1. We had Cho, many few. We had Chan, whatever.
There was no
Chan. All right, so let's look at this. I'm going to go back one so we can look at the nucleotide. What elements do I have? So we have phosphorus. But what are the three we have to have always? Let's start with that. Carbon, hydrogen, oxygen. Always have to have that. Now, what else do we have?
Nitrogen.
I heard nitrogen,
Phosphorus.
phosphorus.
I don't see these so far.
I don't see any. I don't see any S's, do I? So this is just Chan.
Can I be conch instead?
Yeah, Ponch, please. That's like Poncho.
You know what?
Yeah.
If it makes you understand it better, write it down like that. And then paunches, right? If you have a better way to write it down so you remember it, please do. Please do not paunch your lady.